tissues, and basically enter into a symbiotic relationship. So essentially just a nutrient exchange. Plants will give sugars made photosynthetically. In exchange, the bacteria will give compounds from the environment, nitrogen compounds, metals, etc. Things that are difficult for the plant to get. Best studying, or one of the best studying of plant growth promoting bacteria would be the rhizobia of legumes, like was mentioned in the last presentation. The rhizobia will enter into the root tissues of the legume plant and form these root nodules up here, full of bacteria that are fixing nitrogen for the plant from the atmosphere. So rather than looking at legume plants, in Sharon's lab we look at poplar and willow. And the reason why we look at poplar and willow is if you take a look at this picture, it's from the Snoqualmie River, out east. I'm sure most of you are familiar with it. Basically, there's poplar and willow just growing in rocks, which is kind of counterintuitive. Doesn't really make very much sense. There's not much organic matter beneath the rocks. There's not much microecosystem. The river is not very nutrient rich. It's not downstream in agriculture. Yet these plants are still able to grow. And so it begs the question of how. And the answer is the bacteria that are colonizing throughout the plant. So when we isolate these bacteria and we re-inoculate them into sterile plants, the growth differences are absolutely enormous in comparison to uninoculated controls. More so than just the growth differences, they're also some of the strains are really versatile. So they're not just re-inoculated back into poplar and willow, they can be re-inoculated into grasses or crops, tomato or Douglas fir. And so the broader implications of this is that we can really reduce the cost of fertilizer. I mean, for just nitrogen fixation alone, if you look on the back of any chemical fertilizer that's synthesized, one of the first ingredients is going to be some nitrogenous compound like urea. And that costs fossil fuel, that costs gasoline. So reducing that is great. It would really impact our agriculture. And so my research is looking at basically, the goal is to make a consortium that's multifunctional. So not just nitrogen fixation, but these bacteria have many possibilities to benefit the plant. And so what I really want to look at is more quantifying rather than qualifying. Really being able to put solid data to the problem of saying what is a good consortium for a certain crop. So I was working with 8 different endophytes. I was working with, these are some results from Sharon's lab class. The ones highlighted in red are the ones that were given 3 pluses as good symbionts. So just meaning they promoted growth the strongest. As well as Snoqualmie 117.2 and a few other wild poplar strains as well. So the first assay I did on this was a NIFH assay. So NIFH is a gene or a subunit of nitrogenase, the enzyme that fixes nitrogen gas into a more organic form of nitrogen that's more bioavailable for the plant. This doesn't necessarily mean that the bacteria is fixing the nitrogen, but it does provide strong indication that it can. We need a further assay that I didn't actually have time to do in the southern reduction assay to provide further confirmation. And also, this isn't as much a quantitative assay, which is the problem. I'm still looking for a good quantitative assay. But nonetheless, you can see a strong band here at 380 base pairs, which indicates the NIFH gene is present, which is a good start. Good start in looking further into nitrogen fixation. The next assay I did looking at these and my attempt to quantify was siderophore production. So siderophores are they will reduce the insoluble form of iron 2+ to iron 3+, making it more bioavailable to the plant and to the bacteria. Iron is really crucial in enzymatic reactions. Our heme groups have an iron in the center and they have really unique chemistry. So the chelation of the metal makes the transport much easier back into the plant and the bacteria. And so this is the EDTA, just kind of a little schematic of it. Basically just wraps the metal up and makes it a little bit more easy to transport. So I had a lot of issues with this. I spent a lot of my time trying to figure this assay out. What I wanted to do is try to there's a dye that will basically turn yellow as more iron is chelated. And what I wanted to do is try to analyze it colorimetrically. But I ran into a lot of issues with exopolysaccharide formation because the analysis of the colors is actually at 620 nanometers and cell densities at 600 nanometers. And so extraction became really difficult and it kind of got really murky, all the results did. And so the more research I did into this and more reading, the more I realized that we really need to know more about what specific siderophore is being produced by the bacteria. And that takes a lot of time and a lot of research. So I kind of left off feeling like the qualitative assay was the best. Just the cast agar plates, and it produces a little yellow halo if siderophore is being produced and iron's being chelated. So on to what I was actually successful with. This is IAA production. So IAA is a part of the plant auxin family. It's a rooting hormone. So it's going to allow the plant to be able to spread its roots further underground. It's produced by the plant, but if bacteria will produce it, that's fantastic. So this is actually reported in IA production per cell density. And the reason why I reported this in IA per cell density is because I really think in building a consortia, we're going to want to look at a strain that's producing the most amount of product for the least amount of sugar required. Because really, I mean, we can't bank on the fact that there's going to be an end-all be-all one strain that's going to have all of the factors needed to promote growth. And so we're going to want to leave room inside the plant for sugars made photosynthetically for other strains. So one thing I think is really interesting about this assay is that tryptophan, that it's done with and without tryptophan. And tryptophan is a difficult amino acid to make. It's usually a non-essential amino acid. So I think it's really interesting seeing some of these and the differences between plus and minus. One that can produce IA without tryptophan is obviously more beneficial. But you see here 8-1 and 5-2 are really great IA producers. So the phosphate solubilization. So generally how the qualitative assay works is basically there's a agar plate with insoluble phosphate in the solution in the agar. And it's in the form of tricalcium phosphate. So bacteria is plated onto the plate, and what bacteria will do is lower the pH of the system and of the environment, and it will cause the phosphate to solubilize, producing a zone of clearing around the bacteria. So rather than doing that, we grew it up in picoskaya broth, which is similar as a broth culture, similar to the agar, and then mix it with the reagent after over a period of time to get kind of the same thing I was going for with the Sedera progression, colorimetric differences, and analyze it at 820 nanometers on a photospectrometer. Phosphate's a crucial molecule, crucial for driving enzymatic reactions forward in the form of ATP or GTP, and it's conserved across all life. Which is what's really important in looking at these trend lines. If you see actually right here, this is over a period of 4 days, or 3 days with the first extraction at inoculation. But if you look here at 8-1, this is again reported in soluble phosphate per cell density. But if you look at 8-1, the amount of phosphate solubilized per cell density shoots up by the second day, right here, and then it plummets. And that's not because there's less phosphate being solubilized, but it's because the cell density is going through the roof. And that's because phosphate is crucial for life. And we're going to want something that's producing phosphate and leaving it in the environment rather than using it for its own growth, leaving it for the plant, essentially. And so you can see here with these L5-2 and L7-2 strains, they're producing more phosphate while keeping their concentrations fairly stable. So my plans for next year, I really want to better understand the interactions of multiple strains in vivo. I mean, this is in vitro, so it's not a great picture of what's actually going to be happening. And so I'd really like to more move into how these are going to be interacting in plants. Plan I have for next year is to look at combine multiple strains across genera and look and see whether or not they're symbiotic with each other or competitive with each other. And I think that'll provide, as well as with this data as well, provide interesting information and to really understanding to see if multiple strains are producing additive effects with these quantifying the plant growth promoting factors, or if they're deleterious or not. Thank you. Any questions?
Questions for Jack?
How linear is